Good afternoon, dear viewers. In this lecture, we will be talking about the processes of the cell, the basic concept, what exactly the cell does and what exactly the cell is made up of. So before going into the various processes of the cell, we should discuss about the basic unit of the life, what cell is. So all the organisms are made up of cells. So cells are basically the simplest collection matter that can live. All the cells are related to one another and they perform some characteristic and specialized functions. Our body is made up of a number of different kind of cells which are specialized to functions in various different ways. For example, the cardiac cell, which helps for the heart functioning processes, then the nervous cells or the nerve cells, which helps in the transmission of the nerve impulses, then the muscle cells, which helps in the regulation and the locomotions of the muscle cells. So the cells forms the basic unit of life and they are the most important integrity for the survival. Now there are different types of cells and each of these cells play a vital role and a characteristic role for the growth, development and day-to-day maintenance of our body structure. So if we see over here, some of the pictures are being shown of these cells, human cells, which are stained with various different stains. So in the first one we can see the nasal cells which are stained over here and it is watched under the light microscope. Then the second one are the onion cells which are also watched under the light microscope. So we can see in this cell it is surrounded by a cell membrane and inside this cell there is a prominent nucleus which plays a very important role in maintaining the development and the integrity of the cell. Now this is a timeline of the various sizes of various structural entities. So we can see over here this is a timeline which shows from 10 meter to 0.1 nanometer. Now over here if we see there are different molecules which are present showing of different sizes. So we can see there are proteins and lipids which fall under in the category of 10 nanometer to 1 nanometer in size. Then there are bacteria, the smallest bacteria, viruses which are of a size of about around 100 nanometer. Then comes the various bacteria. Then there are nucleus and the various cell organelles which comes into the play, which are present in a size of about 100 to 1 micrometer, and then the egg sizes comes into play. Now these particular cell organelles are can be observed under the light microscopy, and if we want to figure out or position them in a better way, we watch them under the electron microscope, and there are various electron microscopes which are available, like the scanning electron microscopy, there is transmission electron microscopies which are also there. So we can see over here from this particular diagram how exactly the sizes of the various kind of cells, organelles, proteins, lipids they are varying in their structure. Now if we trace back the history of the cell and how exactly these were discovered and they were first identified through the history. Now in the 1665 Hooke was the first one who can identify and name this cell. Subsequently Leeuwenhoek in the year 1674 observed the living cells and could see the greater details due to better lenses in his microscopes. Now subsequently Sheldon and Swan they came up with a theory which was called as the cell theory. So they noted that the plants are made up of cell. Subsequently they concluded that all living cells are living things are made up of cells. Subsequently to this particular cell theory there was an addition of a point which was given by Vacuole in the year 1855. He proposed that all cells comes from the pre-existing cells or the other cells. So this formulated the modern day cell theory concept. Now if we see, the first cell was observed by the scientist Robert Hooke in the year almost 1635 to 1703. Now the word cell was coined by him when he observed under the microscope a cock cell. So we can see in this image over here, we can see the cock cells which are looking like the honeybee comb and which was observed under this particular microscope, and he found that they appeared to be blocks of material making up the cock. So he was the first one to name these or identify them as the cell. Now subsequently he wrote in the year 1665 that I took a good clear picture of the cock with a pen knife sharpened as keen as a razor. I cut the piece it off and then examined under the microscope, and I could perceive it appears to be a little porous, much like a honeycomb. So if we see the image over here, we can see the cock cells which are attached to one another, looks like a honeycomb. So Hawke for the first time called these porous cells because they reminded him of the cells inhabited by the monks living in the monastery. So that's how the name the cell came into the picture. Subsequently, in the year 1838 and 1839, there were two scientists, the Schlender and Schwann, who started working on the cell concepts, basic concepts of the cell and the cell theory. So Schlender was a German lawyer who turned into a botanist. So he, despite of differences in the structure of various tissues, he proposed that the plants were made up of cells and that the plant embryo arose from a single cell. So for the first time he can propose that the embryos arose from a single cell. In the year 1839, Schwann, a German zoologist and a colleague of Schlender's, he published a comprehensive report on the cellular basis of animal life, in which he proposed about what exactly cells are and how exactly they are formed and forming the basic unit of life. Now, the observations of the Hawke, Leeuwenhoek, Schlender, Schwann and Vacuole led to the development of the cell theory. So the cell theory is one of the theory which is regarded as the most explained theory between the cells and the living things. So the cell theory is basically conceptualized into three major points. The first one says that all living things or the organisms are made up of cells. The second one is new cells are created from the old cells, divided into two processes. And the third one is cells are the basic building units of life. So these formulated or formed the fundamental structure of the cell theory concept. Now, if we see the Earth biogeologic clock, which is a portrait which is shown over here, which is a portrait of the past 5 billion years of Earth, we can see how exactly the origin of life took place and how exactly the life, the photosynthetic bacteria, then the cyanobacteria came into the picture, eukaryotic, algal kingdoms came into the picture, then the vertebral vascular plants came into the picture, mammals and the humans evolved through the various timelines. So this is a Earth's biological and biogeological clock which is showing how exactly all these particular structures got evolved from the cells and the cell origin. Now, it is being said that the first cell appeared, the life first emerged at least about 3.8 billion years ago, which is approximately 750 million years after the Earth was formed. Now, over a period of time, there has been lots of speculation how exactly the life originated and how the first cell came into matter, because there were lots of speculation that the initial environment was not so suitable for the development of this cell like the environment what we have. So there were lots less of oxygen, more of carbon dioxide and the other gases like the methane and the other gases. So how exactly these formed were not clear. Subsequently, there were lots of experiments which came into the picture which gave the scientists a clue how exactly these cells were developed. Now, one of the instrumental and the most important experiment which was done by the Uri and Miller experiment, and this was an experiment which proposed that how a chemical experiment stimulated the condition which was created in the early environment, and through that particular thing they created that how the chemical origin of life got created under the primitive condition. So if we see the experimental setup over here, the experimental setup was made in which there was a huge glass chamber which basically contained the primitive condition gases, which were mainly the methane, ammonia gases, water was there, hydrogen gases were there. So there was very less or absence of oxygen which was present over here. Now, in the U-shaped tube, what was there were under the heat conditions and the the electrode spark, what happened is there was creation of certain biomolecules which forms the fundamentals of the earth life cases. Like the organic molecules, like there were formation of number of amino acids like the alanine, aspartic acid, glutamic acid, glycines were formed, urea was formed, lactic acid, and the various other acid which form the fundamental structures for the development or the creation of the first cell was created over there in the primitive conditions. So Uri and Miller experiment proved to be fundamental and one of the most breakthrough experiment which gave us the clue that how exactly in the primitive conditions there was production of the various biomolecules which are important for the cell survival and cell sustainability. Now, the cells have been broadly divided into two major categories. One is called as the prokaryotic cells, the other one is called as the eukaryotic cells. Now, there is a structural and a functional difference between both of these particular cells. Now, the organisms in the domain of bacteria and the archaea falls under the category of the prokaryotic cells, while in the case of the eukaryotic cell, the protists, fungi, animals and the plants, they come under the category of the prokaryotic cell. Now, the prokaryotic and the eukaryotic cell are differing from one another based on a number of characteristics. Let's take some of the characteristics over here which are represented in the tabular representation. Now, if we see in the prokaryote, there is an absence of a nucleus, while in the eukaryotes there is a presence of a nucleus. The typical cells in the prokaryotes is about 1 micrometer, while in the case of the eukaryote it is about 10 to 100 micrometer. The cytoskeleton is absent in the prokaryote, while in the case of the eukaryote it is present. The cytoplasmic organelles like the Golgi complex, mitochondria and the various organelles are absent in the prokaryotes, but in the case of the eukaryotes they are present. So, the eukaryote cells are very specialized and they are compartmentalized to perform the each and every activity in a various different compartments. So, there is a compartmentalization and specialization of the corners within the cell. Now, the DNA content, if we see in the prokaryotes, is less, while in the case of the eukaryotes is a very large quantity. While the genetic material or the nucleic acid which is present in the prokaryote, it is generally the single circular DNA molecule which is present, but in the case of the eukaryotes, it is multiple linear DNA molecules which are present. Many a times in the eukaryotic cells, they are formed in the form of the chromosomes also, which we refer as chromosomes in the eukaryotic cell lines. Now, if we see the basic characteristics of the prokaryotic cells are characterized by having no nucleuses. So, there is no nucleuses which are present in the prokaryotes, while there is DNA which is present, but it is present in an unbound region which is called as the nucleoid. Now, most of the organelles which are there is no membrane bound organelle which are present in the prokaryotes, while the cytoplasm is bound by the plasma membrane. So, the prokaryotic cells are very primitive in nature and they don't have a specialized nucleus or a membrane bound organelles which are formed over here. Now, if we see these are the thin section image of the bacillus. The image on the right hand side is a transmission electron microscopy image of the bacillus. So, we can see over here a bacillus cells which has got a flagella which is present. Now, if we see the diagrammatic representation over here, there is a presence of bacterial chromosome which is basically the singles which is a DNA, circular DNA which is present over here, but there is no presence of any nucleus in which the DNA is present. So, it is present only in the nucleoid, while the ribosomes are present, the plasma membrane is present and the cell wall is present. So, there are absence of any membrane bound organelles which are present in these particular prokaryotic cells. While if we see the structural representation of the eukaryotic cells are much more specialized. So, the DNA in a eukaryotic cell is present within a specialized organelle which is called as the nucleus, which is bound by a nuclear envelope. So there is presence of membrane bound organelles which are also present, and subsequently cytoplasm in the region present between the region of plasma membrane and the nucleus. So between the plasma membrane and the nucleus there is a presence of a region which is called as the cytoplasm. Now, if we see the structure over here, this is a cross-sectional structure of the eukaryotic cells, which majorly consists of the protist, fungi, animals and the plants kingdoms. So, let's discuss about the structural details of these eukaryotic cells. So, this is a structure of a eukaryotic animal cell which is shown over here. So, we can see there is a presence of a outer membrane which is called as the plasma membrane. These plasma membrane are basically made up of lipid bilayer. So there is a lipid bilayer which is present over here. Inside the plasma membrane there are presence of various membrane bound organelles which are present, which we can see over here: the Golgi apparatus, the ribosomes, lysosome, mitochondria, peroxisomes. Then there are various cytoskeletons which are present over here, the microfilament, intermediate filaments, microtubules, which provides the rigidity and stability to the cells. Then there are centrosomes which are present over here. Apart from them, there are endoplasmic reticulums, which is basically the rough endoplasmic reticulum and the smooth endoplasmic reticulum, which is attached to the nucleus through the nuclear membrane over there. Now the nucleus is broadly divided into many different regions. There is a outer nuclear envelope which is present. Within this one there is a chromatin which is present, which is many a times in the form of a dense structure referred as the chromosome. Then there is a nucleolus which is present, which is basically responsible for the synthesis of the ribosomal RNA, and then there are flagella which are present for the movement of these particular cells. So we can see this is a specialized, a typical cell of the animal cells in the eukaryotic class. Now if we see the structural representation of a plant cell and how exactly a plant cells looks like. So plant cells are more or less similar in a way to the animal cell. Only thing, there is difference of some of the organelles which are present especially in the plant cells, because they are responsible for photosynthesis processes. Now the plant cells are covered by additional membrane which is called as the cell wall. So each of the plant cell is attached to one another, or there is a presence of cell wall through which it communicates. So the cell wall has got lots of perforation, transmission pores which are present, which are called as the plasmodesmata, through which they connect to one another. Now inside the cell wall there is a presence of the plant cell membrane, and within the plant cell membrane there is a presence of various organelles which are present. So if we see over here, there are different organelles which are shown over here, like the Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, ribosomes are present. Then there are nucleus which is present, which can be differentiated into various regions like the nuclear envelope, nucleolus and the chromatin region. Now in the plant cell we can see there is presence of a number of organelles which are characteristically important for the photosynthesis, for example the chloroplast cells. Now beyond the chloroplast cell there are presence of cytoskeleton, which are basically the microfilaments, intermediary filaments and the microtubules which are present over here. Now along with these particular organelles there is a presence of central vacuole which is also there, which plays a very important role in the plant cells. Now if we see, this is a typical structure of the plant cells and how exactly it looks like different from the cell. Now the first culture of the human cells began in the year 1951. So the first human cell culture was done, and this was called as the Hela Cell. So if we see the picture over here, this picture is showing the Hela cells which are grown in the petri dishes. It was called as Hela cells because it was taken from a patient. His name was Henrietta Lacks, so the name came into as the Hela cells. The patient was suffering from malignant tumors, and these cell lines were cultured for the first time, and then they were cryopreserved for subsequently use. Now, these cell lines are still being used majorly in the cancer biology cases or study of the various cancer cell lines also. Now, if we see the levels of cellular and molecular organizations within the cell. So, this is a microscopic structure of the villus of the wall of the small intestine. So, if we see the electron micrograph which is shown over here, this is showing a cross section of the small intestine, and the villi are subsequently focused upon. So, we can see how exactly the villi are made up of with specialized cell to bring about the specialized functioning of these particular organization. Now, this is a representation showing the evolution of the cell, how exactly the cells evolved through the billions of years. So, from the first cell, how exactly the cyanobacteria came into the picture, then subsequently the prokaryotic cell lines came into the picture. The chloroplast got endosymbiont into the cell lines, and how exactly the plant cells came into the picture. Then the animal cell lines, fungi, protista, archaebacteria, how exactly they diverged from the various processes of the evolution over a period of time. Now, this is a structural entity which is showing the various organelles which are present within the cell. So, we can see the various organelle, the plasma membrane, Golgi apparatus, nucleus which is there, the mitochondria is there, the nucleus is there, endoplasmic reticulum is there, smooth endoplasmic, rough endoplasmic reticulum, how exactly they are present over here. This is a stained image showing the representation of the plant cells. So, the plant cells, the various types of the plant cells are shown over here. So, we can see the plant cells, how they are formed and how exactly they are stained under the various stains and when they are watched under the microscope. Now, these are the light micrographs which represents the animal cell lines. So, how exactly the animal cell lines look like when they are processed, stained and then observed under the microscope. Now, then what comes major within the cells is the biomolecules which are present within the cells. The major biomolecules which are present within the cells are basically the carbohydrate, lipids and the amino acids which are present. Now, the carbohydrates many a times are referred as the saccharides, which are basically the molecular compounds made up of three elements: carbon, hydrogen and oxygen. So, over here the saccharides have been broadly classified upon various categories. One is called as the monosaccharide, then the disaccharide and the polysaccharide. So, monosaccharide is basically where there is presence of only one sugar or one saccharide is present, which is basically glucose, fructose, galactose. While in the case of the disaccharide, there are two sugars which are attached to one another. In the case over here which is shown is sucrose and lactose which is shown over here. Then comes the polysaccharide, in which a number of sugar molecules like the glucose are attached to one another in a normal chain and then in a branching, which is shown over here as the glycogen molecule. So, the glycogen molecules is having the glucose monomers which are attached to one another by alpha 1,4 and alpha 1,6 bonds, which forms the linear chain and the branching chains in the glucose monomers. Now, how exactly from the monosaccharide the disaccharide is formed? So, there are basic process of hydration and dehydration which takes place for the formation and dissolving of these disaccharides into monosaccharides. So, if we see a picture over here, this is showing a basic example of the formation of sugar which is shown over here. So, the sugar is basically made up of monomers of glucose and fructose. So, the glucose and fructose, what happens is they combine to one another. There is a release of water molecule due to the hydroxyl and the proton which is released from the glucose and fructose respectively, and there is a formation of the glycosidic bond between the alpha 1,4 carbons of the glucose and the fructose molecule. So, the sucrose molecule is formed over here. while the next case is showing the maltose, which is a monomer of glucose which is combined with a glucose. Then there is a lactose, which is galactose plus glucose. So there is various combinations which are formed of the various saccharides, which combine to form various disaccharide, which subsequently combine to form the polysaccharide molecules. Now, the structure of the saccharides are very complex in the cases of the polysaccharide. So, if we see, this is a representation of the glycogen molecule. So, we just discussed about the glucose monomers which are attached to one another by alpha 1,4 bond to form the linear chain, and subsequently there are branching which is taking place between alpha 1,6 glucose monomers. So, we can see over here there is a linear chain of 1,4 bonds which are there, and there are branching of 1,6 linkages of glycosidic bonds which are formed over here. The next important biomolecule which is present is called as the lipid molecule. So, some of the basic lipids are the cholesterol which is present. Then we know about the triglyceride, diglyceride and monoglyceride. So, tri, di and monoglyceride are nothing but a glycerol backbone to which the fatty acids are attached. So, depending upon the fatty acids, they are being named as tri, di and polyglycerides over here. Now, this is a representation of the glyceride structures. So, how exactly they looks like. So, there is a polar head and non-polar tail which is present, which are the fatty acids which are present. So, they play a very important role in the formation of the lipid bilayer or the plasma membrane and the various membranes present within the cell. Now, the last one which biomolecule which is present in the body is called as the proteins. So, proteins are basically the polymers of amino acids which are attached to one another with the help of the peptide bonds. So, over here we can see there are two amino acid 1 and amino acid 2 which are present over here, which are combined to one another with the help of the peptide bond. So, there is a peptide linkage which takes place over here. Now, each of these amino acids are having a basic structure. So, there is a central carbon to which the proton is present. Then there is a amino group which is present on one end, and there is a COH group or the carboxylic group which is present on the other end, and there is a residual group or the R group which is present. So, depending upon what exactly is present in the R group, the amino acids have been classified into various different forms. So, there are basically 20 amino acids which are present. So, these are some of the amino acids which are shown over here. So, they have been classified as hydrophobic, polar, non-polar, like this one. So, we can see all the amino acids differ in the presence of the R group. So, if there is presence of H, there can be methane, there can be some other cyclic compound which is present over there. So, the biomolecules are basically the saccharides, then comes the lipids and the amino acid. So, in this part of the lecture, we discuss about the basic concepts of the cell, how exactly the cells were discovered by the various scientists, and subsequently how exactly they were named, what are the structures which are present in the cells, and the basic units of the cells which forms the fundamentals for the survival, growth and the development of the human body processes. So, with this note, I come to the end of my lecture. Thank you very much.